joining. I'm going to just hi everyone. So I'm just going to start with the introductions for our speaker today, Rachelle. So let me go to her biography. So Rachelle Bester is a passionate scientist with a strong interest in biochemistry. Her academic journey started at the Northwest University in South Africa, where she did undergrad in biochemistry and physiology. So she wanted to understand human body quite well. And she extended her academic pursuits to include microbiology and chemistry, which has also proven invaluable for postgraduate studies. So she completed her studies and an honors degree in biochemistry with a particular focus on human metabolomics. And since then, she's been fueled with a deep curiosity in the intricacies of the human metabolome. So now she's found her way through her master's looking at these marathon studies and now into her PhD level, looking at the human metabolome. So she has a intense interest in marathon running and human metabolome. And she's gained many insights on the metabolic responses under conditions of intense energetic stress that closely resemble those seen during various pathophysiologies. And it's made her realize how much is uncharted territory is out there, and potentially there's a niche of untargeted metabolomics. So while she takes pride in her academic achievements, she sees this as only just the beginning of her journey to make meaningful contributions to the field of human metabolomics. With that, I'll introduce our speaker, Rachelle. I'll give the floor to you.
Thank you very much, Prof. Shane. I hope you guys are all well, and it's a very good morning to you all. So today we're going to look at an untargeted proton NMR metabolomics perspective of marathon-induced metabolic changes and the subsequent recovery thereof. Let's see. Oh no, my slides don't want to go. There we go. Okay, so I'm going to give a very brief background, considering that the discussion of my results will take up most of the time. Marathon running is a significantly energy demanding exercise, and it mainly requires the breakdown of carbohydrates, lipids, and proteins for energy, and the eventual activation of autophagy as a last minute of a last resort to energy production. These incredible perturbations in accompaniment with insufficient recovery results in a significant decline in athletic performance. And along with this, numerous negative health effects have been reported along with the insufficient recovery, with a few examples such as cardiovascular dysfunction, kidney and liver damage, as well as post-marathon depression. Now, I will not give you the definition of metabolomics, as I'm quite sure everyone is familiar with it. However, metabolomics does provide a closer representation of the altered physical state, thus allowing us to connect some of these metabolites that are perturbed from the marathon, as well as their recovery, with the associated negative health effects that have been reported. So in this project, we utilized proton NMR spectroscopy, which is ideal for untargeted metabolomics, especially hypothesis generating research. And we also utilized the miniaturized method, which is ideal for limited sample volumes, which was the case in the current investigation. So just to give you an idea of our cohort and the sampling time points, we took samples directly before the marathon, well, a few days before the marathon, directly after the marathon at the finishing line, at day 1, 24 hours after the marathon, and day 2, which is 48 hours after the marathon. The groups that we will be comparing is our pre and post group to see what the effect of the marathon itself is on the metabolome, whereafter we will look at the recovery confirmation, that this is basically looking at your 24 hours and your 48 hours afterwards, and whether those metabolites did recover to their pre-marathon baseline concentrations. Thereafter, we had a look at the recovery trend of these perturbed metabolites by comparing the post groups to their corresponding 24 hours and 48 hours post-marathon groups. And finally, we also did a interday recovery confirmation to see whether several metabolites would have changed significantly between 24 and 48 hours after the marathon. Okay, so to jump away, this is the metabolites that we identified as significant and quantified. Those that are indicated in bold are the metabolites that we have identified and considered significant. You will see line graphs next to each metabolite, which indicates their concentration changes from pre-marathon to directly post, and then day 1 of recovery and day 2 of recovery. So we'll start off with the carbohydrates. I'm going to break this up into 2 sections. So the first section will focus on your energy producing pathways and how they were influenced by the marathon, whereafter I will look at several detrimental health effects and several metabolites that were linked to these health effects. So first off, we have our carbohydrates, which are preferentially utilized during the first 90 minutes of the marathon. This eventually, around 90 minutes, the glucose and glycogen stores become depleted. This is also commonly referred to as hitting the wall. Now, very interestingly here, we did see a directly post concentration of glucose that is increased, where you would actually expect a much lower concentration for glucose, considering that it has been depleted. So this has been attributed to the downregulated insulin secretion accompanied with the initial depletion of the glucose and glycogen stores, followed by an elevated glucagon and insulin ratio, and this, along with cortisol secretion, resulted in an upregulated gluconeogenesis, an increased glucose concentration, and along with a downregulated glucose uptake. Thus, we saw an intracellular hypoglycemia and an extracellular hyperglycemia. Now, the recovery of these metabolites were quite interesting as well. So you will notice that lactic acid recovered quite immediately. Now, this might be due to the activation of the Cori cycle and gluconeogenesis, which produces pyruvic acid and glucose in an effort to restore the glycogen stores. Now, the pyruvate concentrations, you will notice it gradually normalized over day 1 and day 2. However, these differences here weren't very significant, weren't statistically significant, and only indicated the normalization of the glycogen stores. Glucose, on the other hand, you will notice that at day 1, the concentration was lower than our baseline level. Now, this is also attributed to the utilization of glucose initially to produce more, to restore the glycogen stores, where after the glycogen stores have been restored, glucose normalization could take place. I also have to emphasize throughout this whole thing that dietary intake is a factor that we should consider. This, however, is an overall perspective that we've got from all of the athletes. So after our carbohydrates, protein breakdown is your next source of energy. Now, we mainly saw the breakdown of valine, lysine, leucine, isoleucine, and proline during our investigation. And this utilization of the amino acids was also substantiated by the similar increase in their catabolism intermediates, thus indicating amino acid usage. Now, along with the amino acid breakdown, you have an increased amount of ammonia that needs to be excreted, thus an upregulated urea cycle. And this you can see here with the ornithine concentrations that decreased directly after the marathon. Okay, hereafter, the proteins, ornithine recovered quite quickly, as you can see, seeing as protein breakdown is not a very major source of energy in the rested state. And thus, ornithine recovered very quickly. Our protein breakdown recovered quickly as well. All of our amino acids recovered quite quickly, except for valine, which is a gluconeogenic amino acid, and may play a role in the resynthesis of glycogen as well. Furthermore, let me quickly see. Yes, next up we have our lipid oxidation. And during lipid oxidation, this mainly contributes acetyl-CoA towards the TCA cycle, and was also indicated by the increased acetylcarnitine concentrations directly after the marathon. We also saw this in the increased concentrations of all 3 ketone bodies, as the acetyl-CoA that is being influxed into the TCA cycle becomes so much, it accumulates so much that we see an saturated electron transport chain. Thus, this also connects to our hyperxanthine, which is due to an elevated purine catabolism initially, from when your ATP is broken down to adenosine, and then hyperxanthine as well. Considering our acetylcarnitine, this carnitine plays a buffering role in the maintenance of the mitochondrial acetyl-CoA and reduced coenzyme A ratio via the carnitine acetyltransferase catalyzed synthesis of acetylcarnitine. Here we have it here. This is as a result of this accumulating acetyl-CoA, not only from the lipids, but from the carbohydrates, from your proteins, from all sources, that is pushing into the TCA cycle. This restoration, however, the ATP restoration is quite immediate. You can see that hyperxanthine recovered very quickly. Our ketone bodies also recovered very quickly, indicating a recovered redox balance. And acetylcarnitine recovery also took place very quickly, back to its normalized concentration, which also is in agreement with the restoration of our high mitochondrial acetyl-CoA and the reduced coenzyme A concentrations. Thus, there is no longer such an intense need for ATP production at this stage. As an interesting thing, this is now where I'm going to focus specifically on the health effects that we noticed from a few of these a few of the metabolites that we quantified. First up, we had liver damage, which we initially we identified using the Fisher's. After the marathon, we have an increased phenylalanine and tyrosine concentration. Now, this balance that we had here, considering the Fisher's ratio, indicated reduced liver function, not necessarily liver damage, but a reduced liver function during this time. Now, this was also supported by a collaborative study of ours, who reported elevated AST levels. Phenylalanine and tyrosine, however, recovered by day 1, indicating the amelioration of the liver function, which does make sense in saying that it is not chronic or severe in any case. However, along with insufficient recovery, this might become a problem if the athlete continues to take part in numerous marathons in subsequent days without proper recovery. Your liver damage, there won't be time for the proper recovery of that, and thus you might end up with liver damage. Next up, we have the gastrointestinal damage, which is not severe or chronic. However, you will notice that we saw a decreased methanol concentration here. Now, methanol is produced endogenously by the gut microbiota via anaerobic fermentation. This is a normal process that takes place in every human. However, seeing a decreased methanol concentration compared to your baseline concentration was quite strange for us. And we ascribed it to the increased mesenteric ischemia, a decreased gastrointestinal perfusion, and thus a slower absorption of this metabolite into the system, thus explaining the lowered concentration. This also had a slow recovery, which is in agreement with reported health effects of marathon runners afterwards. They call it a runner's, what's the word? When you get the runs, it's a wordplay with the marathon runs, and you tend to have a lot of gastrointestinal problems the first few days after your marathon. And this is also indicated by a slow increase and slow recovery of methanol to the normalized concentration, which did not completely normalize by 48 hours in our study, however. Next up, this one was quite interesting. So we had increased concentrations of ethanol and propylene glycol directly after the marathon. And this did break our brains for quite a while, trying to figure out where did this come from? Could it have been a contaminant, which we eventually ruled out. And after some digging, I found out that a lot of energy drinks contain ethanol and propylene glycol as solvents for many of their ingredients, such as ginseng, for example. And an ineffective clearance of this via alcohol dehydrogenase would make sense why these two alcohols were increased directly after the marathon if energy drinks would have been consumed during the marathon itself. However, propylene glycol and ethanol both decreased significantly at 24 hours after the marathon. And this is mainly due to an elevated elimination due to vasodilation and a rehydration of the athletes afterwards. In addition, the breakdown of ethanol and propylene glycol may also aid in the production of acetyl-CoA via lactic acid here and the Cori cycle. This also plays a role in the recovery of your NAD and NADH balance, as well as ATP and your fuel substrate store regeneration. This might also contribute to the decreased concentrations of these two alcohols. However, we did not think that this would be the major cause of the decrease that we saw. It would would likely be the hydration that has been increased, as well as the increased vasodilation directly afterwards. So next up, and this is my favorite part, is the delayed onset of muscle soreness, that we found a bunch of markers that correlated with this. So as opposed to the steady recovery of the branched chain amino acids and lysine, proline over here decreased at day 2 following its initial recovery at day 1. It's a small decrease that you can see there. Now, this is a link to delayed onset muscle soreness, and it coincides with another collaborative study of ours who saw an elevated hydroxyproline at 48 hours post-marathon. We attributed this to a delayed post-marathon collagen breakdown. Now, this is also linked to your increase in creatine at day 2 over here, as well as, and this increase in creatine is also in agreement with another collaborative study of ours who saw increases in numerous inflammatory markers and an epidermal growth factor. And this then upregulated creatine kinase and plays a function in the regulation of muscle anabolism and repair. The slight reduction over here in citric acid at day 2, at your last stop there, might also be linked to the upregulated ATP citrate lyase activity, which is associated with skeletal muscle regeneration and myofiber differentiation. Another marker that is of interest here markers are tyrosine and phenylalanine, who also both decreased at day 2 following their initial recovery at day 1. So this has to do with the expression of guanosine triphosphate cyclohydrolase 1. It's this one over here, which is upregulated by delayed onset muscle soreness related inflammatory markers, the same inflammatory markers that our collaborative study identified. And the upregulated tetrahydrobiopterin synthesis that you have here leads to a upregulated tyrosine hydroxylase and phenylalanine hydroxylase, these two here, activity, thus also attributing to the decreased concentrations of tyrosine and phenylalanine. Another possibility is that tyrosine and phenylalanine may be subsequently broken down and might increase your dopamine epinephrine, and norepinephrine levels, which have also been implicated in the modulation of muscle pain. However, these studies are very rare and there's not a lot of those, so this is completely hypothetical. And yes, that's it. My story is quite short. I do apologize for that. Are there any questions? And thank you very much for your time.
Thank you, Rachelle. So I'm going to open up to the audience. If there are any questions, you are welcome to type in the chat or you can raise your hand. Also, Rachelle, I encourage our speaker to share their email as well if you're willing to do that. So if anyone has any questions later or watching the video later, then they can contact you and communicate with you.
There we go. I've added to the chats.
Okay, great. So in the chat there, that's Rachelle05.rb@gmail.com. So you kind of condensed your whole past 5 years into 15 minutes.
Yeah, that's basically what happened. Yes. It's been a very panicky few weeks after work.
No, it's a very good summary and very interesting results that you have obtained from your study. So
Yes.
my question to you would be, what's next for Rachelle?
What's next for Rachelle? Well, I think you do know what my next aim is.
Well, tell us.
It's seeing whether beetroot juice would have an attenuating effect on these recovery metabolites. We'll see what happens there. We might also, we're talking about publishing another paper. You would be aware of my honor student who looked at the differences between the male and the female marathon-induced perturbations. So by taking this further and also including the recovery time points, we might get a very interesting picture of the difference in how females and males regulate their energy expenditure during these races, as well as their recovery afterwards.
I sat through that honors presentation, and he was very careful to pick his words. He mentioned difference in sex and to not bring in the term gender, because at the moment in our current society, the word gender is very complicated.
It's a very, I warned him about that in the beginning.
So yeah, sex, male and female, that's what he looked at. It was also very interesting results. There is a question.
Yes, I see that.
So Dr. Koji, who presented for us previously, so thank you for the great presentation. Do you think that the differential metabolites associated with amino sugar metabolism are as produced as a means of response to stress, because notice similar results in bacterial culture studies? So
this might actually be a possibility, and I honestly would not doubt that it is something that would happen. We have seen in the previous study where we looked at the in-depth influence of the marathon itself. We did see a lot of metabolites that were influenced by the stress itself, by hormones excreted during stressful situations. So yes, I would actually, if you have a study published on that, I would love to have the reference and have a look at that.
So Piet also has a comment there that you would focus on lactate clearance, because there's a big issue for cyclists as well, and Piet is very much into cycling.
Yeah, lactate clearance, it was a big part of the research for this previous one, and it eventually with the opinions, it's not the right word, the help, there we go, the help from all my supervisors, lactate clearance was basically just reworded or reconsidered as the influence of the Cori cycle afterwards and during the marathon itself to produce additional energy.
And Dr. Korji has a second follow-up question. So how did you decide the control for the study, the participants before the marathon or independent subjects? You might have
Okay,
missed
no.
that.
Yes, so I did not mention this. I didn't go into details regarding our participants and how they were chosen, and I mainly just focused on the results and the discussion of the study itself. So each participant did serve as their own control. We did a paired analysis for this in an effort to kind of circumvent the differences, the genetic differences, and the interpersonal differences that we have in with the metabolomes and the profiles that we saw after the marathon. So yes, we utilized the same participants throughout the whole study, and they served as their own controls. This is why we took the pre-marathon samples a few days beforehand in a fasted state. There we go. Let me just include that as well.
So Rachelle, if you had to refine your study design, now that you've done all of the analysis, all the complications, 5, 6 years later, how would you improve upon, how would you redo this with improvement on your study design?
I would definitely include more participants. We only had 30. And I would have loved to see a wider perspective of the changing metabolome with more people included. I would also stretch out the recovery time points that we analyzed, seeing as delayed onset of muscle soreness only peaks at around 48 hours. We might have missed quite a lot of that recovery in the subsequent days. So by adding a few extra days afterwards from recovery would also have provided a lot of extra information.
And would you want any additional type of samples, or we just stick with blood samples?
Oh, no. Urine samples would be perfect, actually, along with this, just to get a bigger perspective of the changing metabolome. But on the overall, urine and serum would have been the best. I do believe that a lumbar punch would be extremely invasive, although it would have given us quite some interesting snippets on the post-marathon depression.
Yeah. I don't think anyone will want to provide that willingly, but that would be interesting.
No, I also don't think so. Definitely not.
You can even ask them for the bacterial components. The Shane mentioned earlier, Shane Red.
Wishful contribution.
You mentioned runners' trots that you were talking about. Maybe a fecal sample also give something on that.
Sorry, please say that again.
So right in the beginning, while you're doing your presentation, Shane Red said what you were referring to was runners' trots. Well, the
Yes, that's the
methanol,
word.
the gut problem. So getting fecal samples would be interesting to see what is the effect on the metabolism in the gut.
That's true as well, yes. And having a look at your gut microbiome. But that opens up the door to a lot more that I would like to do. That won't be an option right now.
So yeah, there's a lot for Rachelle to come up. Well, I think I'll approach you when you get some more solid results on the beetroot as well, and maybe you can present to us on that.
Yes, that would be lovely. Seeing
I
the
see
whole picture together and how they all interact with each other.
Malati has a question.
Yes, my question is more of a I guess a basic but long-term question about marathon running. So if you become a habitual marathon runner, say you run 4 or 5 very long ones a year, what sort of long-term metabolites do we anticipate should be at a good level? You know, like there are those stress changes, and I'm assuming
Yes.
if running is part of your lifestyle change, what sort of good metabolites do we want to be in high levels?
Well, I would specifically to be able to thoroughly answer that question, I would like to include multi-platform information, seeing as the NMR is not very sensitive. So the metabolites that we do have only gives us a very big outline of what did happen. However, what we have seen with professional runners and but the elite, very intense runners who do not really commit to their recovery afterwards, doing these races one at a time, they generally end up with cardiovascular problems, quite a bunch, as well as renal and liver damage. This is a very big problem for the elite intense runners, if you can state it like that. But those are very few cases, and these are the very highly motivated, highly intense runners that you get.
Okay, thank you.
So there's no real positive metabolites that pop up, just negative?
No, there might be some positive ones. However, we didn't focus on them per se. The metabolites that we saw were all correlated with the negative health effects, and sorry, I lost my words there for a moment. Now, the metabolites that we saw were only correlated with negative health effects, although there is literature on the positive effects of the overall running. It is good for your it's good for psychological well-being, as you know, weight regulation, and surprisingly, actually, cardiovascular health, if it is accompanied, as I mentioned earlier, if it is accompanied with sufficient recovery. You do have this. However, I'm not sure what the metabolites are that are associated with that and how you would see a healthier metabolome of experienced marathon runners. Seeing as our study participants weren't experienced at all, we had a few slightly experienced and a few that it was their first marathon run ever. So we didn't really have anyone in the cohort to be able to look at the positive long-term effects. However, that would be an interesting thing to look at in the future.
Yeah, because I was thinking about when someone is pre-diabetic and there's these high levels of blood sugar. And I was curious about the opposite end of that, where you might have exceeded the normal levels, and you need to watch out for that if, say, you're on the opposite end of that.
Yes, exactly.
Yeah,
that's also something that I've been thinking about. However, there's no research on that, so I didn't really have a lot to substantiate my research on and my hypothesis over there. However, I do agree with you. I do believe, however, that long-term running with the sufficient recovery would result in a decreased insulin, an increased insulin sensitivity, which might circumvent the glucose increase. However, I'm not sure at all.
Okay, thank you.
So I think a good thing to say is sometimes less is more. So endurance running means going beyond what your body can handle, and then pushing it further until it eats its own organs. And if you don't supplement, if you don't treat yourself afterwards, it will continue to eat the organs and hurt you. So you have to be aware that it is a very traumatic experience for your body.
Yeah, continual damage. If you do not recover in between. Honestly, this is what we have seen. A lot of articles, they highlight the positive effects. However, these athletes are also those that have proper recovery sessions afterwards. They do not reach functional overreaching, which is something that I didn't touch on here. It wasn't the specific focus. However, it is an eventual significant decline in athletic performance if there isn't sufficient recovery, which may take between months to a few years to recover from.
Well, there's some food for thought.
Yeah.
So are there any other questions from the audience? Thank you for the questions that were given, Dr. Cordy and Piet. If there are no other questions, I'm going to thank our speaker very much. Rachelle, thank you so much for presenting your findings that you have uncovered just with NMR metabolomics. And we look forward to your beetroot supplement results to see how does beetroot contribute, the new what's it, the new superfood beetroot, how does that contribute to recovery?
And whether it does, yes.
Whether it does, that's the question.
It's a very good question.
Okay, so thanks very much.
Yeah,
And
thank you.
I wish you all the best with your future research.
And same to you guys. Thank you very much for giving me your time.
No, thank you.
Thank you.